Sorry for waiting. Thank you for giving me the opportunity to talk about this topic. And I would like to talk about the genome assembly and polymorphism analysis of a plant conducted in our research institute. So first, I'd like to introduce our research institute, Kazusa DNA Research Institute, which is called KDRI. And the KDRI is located on Kisarazushi in Chiba Prefecture, and it takes 1 hour from Tokyo Station by taking a bus. And our research institute is surrounded by this forest. And this KDRI has established in 1994, and it has large, mainly two research groups related. Sorry, KDRI has two large research groups related to plant and human genome, and in the plant genome research groups, we applicate the genome and the gene information to plant breeding efficiently, which is called genome-assisted breeding. And we use this information to cultivar identification. And I belong to Facility of Genome Informatics, and we are doing the bioinformatics researches such as genome and transcript analysis and polymorphism analysis, and we are making many kinds of databases. And KDRI has these NGS sequencers and short-read types and long-read types. In short-read sequences, we have high-seq data which is released from Illumina. And as for long-read sequencers, we installed the SQL last year, and we have Minayon released from Nanopore. Now we are mainly use this SQL for genome sequence of plant species. And this here shows the computational system in KDRI, and we the data from sequencer was transferred into the storage system, and we have this large storage system whose total amount of the storage is about 10 petabyte. And we have shared memory servers which has a large memory size in each node, and totally we have 20 terabyte memories. And we use these servers for assembly of the large size of genome sequences. And we have cluster servers whose features composed of 63 nodes, and total CPU was about 1,000 cores. And we use these servers for blast search or read mapping and so on. And by using these servers, we analyze this sequence data, and the real result was released from the web server. So we make many kinds of databases. And here is the genome sequences whose sequence was determined by KDRI. And we determined this plant genome of plant species. And here shows the diploid chromosome structure. And diploid chromosome structure has 2 haploids. And we determined these haploid sequences. And according to the similarity between 2 haploids, we classify the plants into a low heterozygosity 1 and high heterozygosity 1. And in the case of low heterozygosity plant, we have determined at first Arabidopsis thaliana in 2000 with international collaboration. And after that, we determined Lotus japonicus. Lotus japonicus is a model plant of beans. And after that, we determined the genome sequence of crops, many kinds of crops, such as jatropha and tomato, and eggplant and sweet cherry, and so on. In the case of the genome of low heterozygosity, the assembly is not so difficult because the genome structure of them are not so complicated. And in the case of high heterozygosity plant, we have determined the genomes of carnation and buckwheat and wild roses and so on. And in the case of the high heterozygosity, if we choose the appropriate assembler, the assembly is not so difficult because in the case of so we should select the assembly method to determine the genome as haploid or diploid sequences. And in the case of polyploidy, now we have determined the genome sequence of strawberry, which is octoploid, and now we determine the genome sequence of sweet potato. It is hexaploid, and it is ongoing. And the assembly is very difficult for the plant species of polyploidy because the chromosome structure is very complicated. And the effective assembly method for polyploidy organism is not so determined yet. So I'd like to talk about the genome sequence of medicinal plant, and it is ongoing. And this plant is, we consider that this plant is very important because these plants produce anti-cancer metabolites. And it has diploid chromosomes with high heterozygosity and 11 chromosomes. And genome size of this plant is estimated as 430 megabases. And we obtained the PacBioRes by using RS2 and SQL. It's a long read type sequencers for 16 and 3 cells, and collected them, and the total amount of collected read coverage is about 4x. And here shows here is a genome assembly conducted to the PacBio data by using Falcon and Falcon ANSI. It's a software name. And in the Falcon assembly, the PacBioRes were assembled into this initial assembly graph. And first, we construct the contigs, including SNPs, and as common regions. And in the variable region, such as structural variant, the contigs build bubble structure. So we first construct this initial assembly graph, and the next step, we select the primary contigs we select the common regions and one side of structural variable region, and we get long primary contig. And remained short contigs were separated, and they called as associated contigs. And this is a result of Falcon assembly, and here shows the result of this. And we get 383 primary contigs, which is long contigs, and the total size was 442 megabases. And this is close to the estimated genome size for 130 megabases. And N50 is N50 is means the connectivity of the primary contigs, and it was 6.3 megabases, and we consider this result is good one. And in the case of associated contigs, this short one, the total length was 2.0 megabases, and N50 was very short. It's 45 kilobases. We determined the genome structure like this. And next step, the PacBio leads were mapped onto the primary contigs, long contigs, and the PacBio leads were divided into each haploid sequences. In the Falcon, this shows the method of Falcon and zip assembly, and extend the sequence, variable region sequence, including SNPs and structural variations, to connect to construct two haploid as like this. And this sequence colored in blue means primary contig, and red means haploid. Haploid is extended by using associated contig and intermediate common sequences between the SV regions. And to determine two haploid sequences is called phasing. And this is a result of phasing, and we get long primary contigs and many sequences of haploids like this. And this is a result of Falcon and zip, and the total number of primary contigs was reduced to 84 sequences, and total length was, and N50 was almost same. And haploids was increased, and total number was increased to 130 megabases, but N50 was not different from this result of Falcon. And the next step, we try to extend the result of these contigs, primary contigs, by using Hi-C method. Hi-C method is an experimental method and used for scaffolding primary contigs. Hi-C means chromatin conformation capture sequencing, and by using this technology, we can obtain the long-range sequence, pair-end sequence. And this is a method of Hi-C, which is developed by Phase Genomics. And first, histone and fragment DNA was mixed and constructs cross-link DNA like this. And next step, cut with the restriction enzyme for these DNA sequences, and attach the biotin and ligate them. And next step, we share this DNA, and we pull down the fragment DNA with biotin and sequence them by using elimination lead sequencers for pair-end leads. If we use this technology, we can get ultra long range of insert size, and it is said that the insert size is up to can be covered up to 30 megabases. And if we use this ultra length pair-end sequences, we can connect the contigs to build more longer sequences. So we apply this method to our result, and the assembly by using Hi-C data is done by using Falcon phase, and this Falcon phase is called the haploid assembly method. And this figure shows the procedure of Falcon phase. And first, we use the result of Falcon ANSI. And in this step, we use high-C data. And first, we construct a phase block, and they are connected by using high-C long-range paired with these, and connect for the phase block and primary contigs like this, and we construct the full length of primary contigs two long primary contigs. They are called phase 0 and phase 1. And this is a result of the Falcon phase, and we finally get phase 0 primary contigs for 184 sequences, and total length was 441 megabases, and MCT was 6 megabases. And the other side of haploids, we get phase 1 sequences for 184 sequences, and total length was almost same, and N50 was almost same. And we merge these sequences, and finally we get 368 sequences. Its total length was 881 megabases. And we succeed to construct these two kinds of haploid sequences, like this diploid structure. By using this high-C data, two long haploid sequences can be constructed. And next, but this result has many primary contig, many sequences. So we try to extend by using BioNano data. And this BioNano is use optical map like this, and fluorescent label was attached to the restriction site in DNA, and construct this optical map, and according to the restriction site in DNA was aligned, and construct this consensus map. And this consensus map has information of restriction site of enzyme, restriction site. And here shows the result of the consensus map of BioNano, and here shows the contig sequence which is assembled by sequencers as sequence read, and they have restriction enzyme site. So we align the restriction enzyme site onto the consensus map, and we get the so we connect the contigs, and this is a final result of this BioNano, and we can get 16 sequences, and the total size was 439 megabases, and the MCT was extremely increased and 38.3 megabases. And this medicinal plant has 11 chromosomes, so the number of the pseudomolecule was 16, so we determined these sequences as like chromosomes. And this is a result of the medicinal plant assembly, And here I talk about another plant by using 10x genomics data. And this assembly is also called haplotype phasing. And this technique first fluorescent labels were attached to the DNA, and here is filled by oil, and DNA and fluorescent label was enclosed by oil, and the DNA in we first gem code, in small character. We make the gem code library, and we amplify the DNA in the oil drop by using amplified, and sequence them by using Illumina short-read sequencers. And if we get about 50x coverage for these Illumina reads, we can assemble by de novo assembly. We can conduct the de novo assembly, and we get the phased chromosome structure. So we tried two kinds of plant species by using 10x genomics data, and this is a result of them. And in the case of plant 1, which estimated genome size is 2 gigabases, and the total size was, this is the result, and the total size was about 4 gigabases for 2 haploid sequences, and N50 was about 600 kilobases. And this is, I think this result not is good result. And in the case of plant B, which has 10, in this plant B, the estimated genome size is 10 gigabases, and the result was total size was 10 gigabases, but the N50 length was only 100 kilobases. And this is not so good result, because the supernova has a maximal input read size, and its size was 2000 megareads. So this amount is not enough for assembling for de novo assembly of this plant species, which has 10 gigabases genome size. And here shows the method for haploid assembly. As I mentioned, this is a backbio approach and 10x genomics approach. And I think it takes high cost for backbio approaches, and it takes $48,000 to determine this medicinal plant, but we can get high accuracy genome by using Falcon and Falcon Antip and Falcon phase. And in the case of 10x genomics approach, it takes a low cost. It takes $6,000 for this medicinal plant, but it is not so expensive. But the result is there is many scaffolds obtained, so the quality of the result is not the quality of the assembly result is lower than the backbio data. And there is on the other hand, there is an gene. It's a company name, and in Israel, company in Israel, and this company has a de novo magic program, which is used for de novo assembly, and but this software is not released. So the assembly method is well unknown. So, but they produce a very high quality assembly result, and it takes extremely high cost. In the case of the high heterozygosity plant with 1 gigabases, it takes $46,000. And in the case of, this program can apply to the polyploidy organisms. And in the case of polyploidy tetraploid with 1 gigabases, it takes $90,000. So we think this assembly method is very high, expensive. So many researchers try to the BacBio and the Genome approaches, and we also apply this assembly for many kinds of plant species. And so I'd like to move the topic to polymorphism analysis in plant, and here I would like to talk about the polymorphism analysis among tomatoes. And the tomato genome sequence has been determined in this year for the cultivars. The cultivar name is Heinz, and Heinz is a company name of ketchup sauce, and the genome sequence was determined. And it is said that about 8,000 cultivars in the world, and they were differed in forms like this, and colors, and fruit sizes, and taste and flavor, and so on. So we want to breed the tomatoes with elite trait, such as disease or stress resistance, and good taste, and so on. So we try to do the two approaches by using sequencing and base array. So first, I'd like to talk about the result of base array. And we use two kinds of base chip. They were released from Illumina, which is Golden Gate and Infinium. And in the Infinium, more SNPs can be obtained than Golden Gate, and the quality of SNPs is higher than that of Golden Gate. And so Golden Gate is not available in these days, so we can only obtain the Infinium base chip. But we used in this, in this study, we used Golden Gate and Infinium. And basically, in both methods, DNA was hybrid onto the probe on the base array and detect the fluorescence. And we used these 40 tomatoes, and this is a edible tomato. They are including the 227 cultivars, and they were inbred lines and F1 hybrid, and they were used for food. And 5 cultivars of microtom were applied to this study, and they were not used for food. And they were used for a model plant because they have a short generation time and relatively easy to get recombinant. As for wild species, we used 8 species of wild tomatoes, and they were found in mountain or field, and they are non-edible because they have a toxic alkaloid. But it is very important because they can apply to crossing to edible tomato to bring disease or stress resistance, and so on. And this is a result of the clustering of the tomato species applied in this study. And we get the SNP genotyping data by using Infinium and Golden Gate for these SNPs. And we select the high quality SNP data, and we get 7,000 SNPs loci. And by using this SNP genotyping data, we get this dendrogram. And we found that 4 large clusters, including wild tomato and tomato microton and F1 hybrid and inbred lines. And next, we investigate the SNP effects on gene or protein functions. And so we classify the SNPs based on the location around the genes into the 6 categories. And C-SNP is a coding SNP, which means non-synonymous mutation. And S-SNP is the silent SNP, and I-SNP is located on intron, and R-SNP is upstream of genes, and U-SNP is downstream, and G-SNP is located on another region. And this eye chart shows the number of these SNPs, and we found that C-SNP and R-SNP and U-SNP is very large number, including in these SNPs, total SNPs. And we consider that C-SNP and R-SNP and U-SNP is very important because they have probability of effects on gene or protein function. So we focused on the C-SNP. So we investigate the SNP location on protein 3D structure because the SNPs directly affect to the function of protein. So we select the genes having C-SNPs, and these genes were selected, and they were their three-dimensional structure was predicted by using homology modeling, and we succeeded to construct for 758 genes, and according to the structure of this surface structure, we predict the function site of these proteins by using this program. And finally, we find that C-SNP in functional site in 200 genes. And this is one example of the SNP result. And this is a concanavalin A-like lectins, which is called BRICS genes. And it is important. It is important because this protein related to sugar metabolism, such as sugar content in fruit. And this is a 3D structure of this protein in tomato. And this structure is constructed by using the Arabidopsis thaliana, this PDB entry for the template. And this is a result of tomato structure. And we found the functional site as shown this meshed region. And we found that the SNP location, location here, and in cultivated tomato, this amino acid is asparagine, And in the case of white tomato, they have asparaginic acid. So this mutation might be related. We consider that this differences related to the differences of sugar content between cultivated tomato and wild tomatoes. And now we constructed the tomato SNP databases. They have SNP genotyping data and graphical genotyping data and three-dimensional structure, which are modeled by homology modeling, and the SNP location can be found in by using Genome. And we consider that these functional SNPs would be effective for plant breeding. And we investigate the SNP by using NGS data. And here shows the plant species whose SNPs was detected, and we target for these plant species features plant species whose genome sequence was determined in KDRI, and this is a result. This is a plant species determined in another research groups, and by using NCBI SR database in NCBI, and we investing the SNPs for these plant species. And now we are making a database for these plant species. And this is a example of the result. And this is a case of JBrowse, which is Genome browser. And this result shows Lotus japonicus. And the mapping result was shown here, and the difference between GIF and Miyakojima is cultivar name, and the difference between them can be browsed on this JBrowse, and we can get the relationship between the genes and the polymorphic loci, and SNPs and insertion can be browsed by using this JBrowse. And if user click this SNP loci, the user can get the SNP annotation information, such as SNP description and the location of SNP and SNP annotation. SNP annotation is a SNP effect on gene or protein function, as I mentioned. And quality of the SNP can be browsed. And now we are making this database, and this database will be released this year. And the SNP database is located in this marker database, and we construct now we publish the database of plant genome database in Japan, which is released in this URL, which is called PGDBJ. The SNP database, as I mentioned, is located here. And this PGDBJ has three main databases, which is also DBs, which include 40 species, and Plant Resource DB, it contains 6 species and 900 storage resources. And in this DNA marker DB, the DNA marker for 65 species and QTL information can be available. And it has a cross-search system for this database, and the metabolome database, which is knapsack and mass base, which are developed in Japan. And as I told, our research institute activity, and from here, I want to talk about the DNA Analysis Support Center in KDRI. And we have a center for development of biotechnology industry to support industrial use of genome-related information and analysis method. And we have three divisions related to mammalian genome resources and DNA analysis and typing, and metabolite analysis and characterization. And this is a division of mammalian genomics researches. And now we provide the bioresources such as human genetic clones and plant genetic clones and ORF clones or antibodies. And by distribution of these bioresources, the many researchers can conduct immunostaining or expression profiles and in situ hybridization. And here shows email address for inquiries and questions and orders. Now we accept them. And this is a division of DNA analysis and typing. In this division, the genome analysis and gene typing and development of DNA markers are conducted. As for genome analysis, commission, genetic and genomic information can be provided for users. And we provide the DNA sequence such as genome sequencing and the RNA sequencing and iso6 sequence and so on. And we provide library construction such as for NGS library and the backend and GDNA. And we supply the DNA markers such as SNP and RAD-seq and microsatellite and so on. And we can computational analysis for genome assembly or gene finding and the genome comparison in this division. And I also in charge of this analysis. And as for gene typing, gene typing can be performed for organisms based on the several analysis methods, such as detection of recombinant and cultivar identification and seed purity test and breed improvement using DNA markers. This is a email address in this division. And final is a division of metabolite analysis and characterization. This provides a method or data for comprehensive analysis of biocompounds. They provide LC mass analysis and LC-MS analysis. In the case of LC mass analysis, we can provide the peak analysis and database searches and statistical analysis, such as PCA, for basic analysis and repeat analysis. And in the case of GC mass analysis, we can provide target analysis and non-target analysis and comparison between samples for basic and fatty acid analysis. This is the email address of the division. So finally, I want to finish my presentation. We are using this current technology to improve the genome quality, genome assembly quality, and generally genome and gene sequence of many crops will be accumulated, and genome-wide polymorphism data will be accumulated by B's array or NGS techniques, and many databases will be constructed. By using these techniques, we expect to molecular breeding for crop improvement improvement will be promoted. Thank you for your attention.